Thanks everyone for joining me virtually today. I'm really excited to talk to you about some of my recent PhD work where I quantify denitrification following floodplain restoration via the two-stage ditch. And none of this work would have been possible without the awesome members of the Tank Lab at Notre Dame who were integral to getting this project completed. It was a huge scale of sampling. Additionally, a collaborator who was kind enough to help us out in the field, Magda Baroza from the Swedish University of Agricultural Sciences, funding from the USDA RCPP grant, as well as the Notre Dame Center for Environmental Science and Technology and their awesome staff who was very helpful in helping in getting my samples run. Now, as many of you are aware, row crop agriculture is a major contributor to excess nutrients to downstream ecosystems. And this happens when excess fertilizer runs off into adjacent waterways and has resulted in elevated stream nitrate concentrations, especially across the Midwest. Excess nutrients often result in algal blooms, fish kills, losses of biodiversity, and other human health impacts such as drinking water impairment. Therefore, we really need agricultural conservation practices to reduce these impacts on fresh waters, as many argue that fresh water will be our next limiting resource. Now, the restoration effort that I want to talk about today is floodplain restoration via the two-stage ditch, whereby we take these channelized ditches here, very straight and narrow, where water is shunted quickly downstream, and convert them into something that looks like this, where we have these inset floodplains constructed, effectively tripling the width of the stream. The construction of two-stage floodplains allows for improved channel stability. It slows the water velocity during high flow events and in turn increases sedimentation rates and also increases the bioreactive surface area and nitrate removal via denitrification, which is the process I'm going to focus on here today. Denitrification is a process carried out by facultative anaerobic bacteria and is a permanent removal mechanism for nitrate pollution in freshwaters as it's converted to N2 gas, which is most of what the atmosphere is made up of. Now, our study site is Shadow Ditch in Kosciuszko County, Indiana. It's about an hour south of the Notre Dame campus, where we have 3 two-stage reaches, the first one being constructed in 2007, and then the next 2 constructed in 2017 and 2018. This totals 4.1 miles, or 75% of the stream length. In 2007, floodplain construction only occurred. There was no touching of the stream whatsoever. However, in 2017 and 2018, there was both floodplain construction and stream dredging to adjust the grade, which gave us a unique opportunity to explore the impact of dredging on denitrification. Therefore, our study objective here was to identify potential time lags in nitrogen processing by quantifying the impact of both stream dredging and two-stage floodplain construction on denitrification rates. We conducted our experiment using sacrificial MEMS, or membrane inlet mass spectrometry assays, by collecting pooled stream and floodplain sediments from multiple reaches and locations throughout the stream. We then homogenized these soil and sediments, weighed them into 50 ml centrifuge tubes, and conducted sacrificial assays, sampling every 2 hours for 8 hours here. And these samples were collected in gas-tight exitainer vials and then run on a MEMS to measure dissolved gases, including dinitrogen gas as well as oxygen. What we were then left with was something that looked a little bit like this, where we have time here on our x-axis, dissolved N2 on the y, and this significant positive relationship through time indicates that denitrification is occurring, and the slope gives us our denitrification rate. To jump into our research questions, the first one that we sought to explore was, how does restoration via the two-stage ditch impact denitrification on stream sediments and floodplain soils? We hypothesized 2 possible outcomes of two-stage construction, including first, system stability, whereby construction occurs and we observe high denitrification rates following construction because these organic matter-rich sediments and soils were exposed to very high nitrate water via the construction process. Alternatively, we hypothesize that we may observe a system reset whereby low denitrification rates are observed following construction because there was a significant removal of organic matter and the microbes were sufficiently disrupted during the construction process. I want to start off my results here by talking about the impact on stream sediments. You'll see two figures like this where we have organic matter following in the black dots across the panel. On the top here, the top bars are our denitrification rates, and the bottom bars are going to be respiration rates. And what we observed was that stream sediment, organic matter, and process rates were not significantly impacted by the two-stage construction process. We hypothesize that this is likely because nitrate is not limiting at shadow, as the concentrations range from about 4 to 8 milligrams per liter, providing a sufficient substrate for denitrification to occur. Additionally, we think substrate type at the bottom of the stream may affect the results we see here, whereby fine sediments are frequently disturbed and the microbial communities may be a little bit more resilient and stable as a result of this. Additionally, fine sediments also enhance anoxic conditions by reducing oxygen penetration into the stream bed. Therefore, our results suggest that in high nitrate systems that are dominated by fine sediments, they may be more stable and be able to bounce back more quickly following dredging in the stream. This supports our system stability hypothesis. Moving on to our floodplain results, we're showing you the same figure we have here, but for the floodplains where you've got organic matter, denitrification, and respiration rate. And these results contrast what we observed in the streams, and that floodplain denitrification was significantly impacted by two-stage ditch construction for over 1 year. This does contrast previous work that was done under redox optimized conditions. However, in our assays, we did not force anoxia, and we feel our results capture a more ambient condition following construction. This is because the construction process may have altered the anoxic conditions on the floodplain by exposing previously buried soils to the atmosphere. Additionally, we observed a 1% increase in organic matter about 2 years after construction, which may have relieved carbon limitation of denitrification, which is another substrate required for these microbes to process nitrate. All in all, our results suggest that the recovery of denitrification is longer on the floodplains than in stream sediments, supporting our system reset hypothesis. Now, we've addressed question 1 here. Jumping into question 2, we wanted to understand how denitrification rates on both stream sediments and floodplain soils in two-stage reaches change over time. And to do this, we compared our 3 two-stage reaches and found that, interestingly, denitrification rates were not significantly different across the reaches of varying age, shown here in this middle panel. There was no statistically significant difference. However, what we did observe was that there was a slight increase in sediment denitrification as the two-stage reach got older. And on the floodplains as well, we saw that in the 12-year-old two-stage reach, we observed a significant increase in both percent organic matter and respiration rates in this oldest reach, suggesting that the age signature is most apparent on the floodplains in contrast to the stream sediments. To begin to put our work into context, we wanted to look at drivers of denitrification in our agricultural systems. And what we found was that, focusing on these 2 panels here, that nitrate was a significant driver of both stream sediment and floodplain soil denitrification. However, we observed a high degree of variability in nitrate or in denitrification rates at high nitrate concentrations, which drove us to look at a secondary driver of denitrification, which was organic matter. And we found that percent organic matter was a secondary limiting factor of floodplain denitrification. This does have implications for restoration in that percent organic matter may be impacted by construction or floodplain age, which may in turn limit denitrification rates. To take another step back a little bit further, let's do the math to see how effective these floodplain restoration efforts are in reducing nitrate export from agricultural watersheds. Now, we know that agricultural watersheds export a lot of nitrogen from the system. And based on our findings here, we're going to calculate a daily removal rate of a stream surface area of 22,000 meters squared and an average rate of 18 milligrams per meter squared per hour. We get a daily removal rate for the stream channel alone of 9.6 kilograms per day. This translates to about 3,500 kilograms of nitrogen per year. When we contrast this with the floodplains, we have a floodplain surface area at Shadow Ditch of 22,800 meters squared, average denitrification rate of 19 milligrams per meter squared, which results in a daily removal rate of 10.4 kilograms of N. This translates into a slightly higher annual removal rate of 3,800 kilograms of N per year, but this is considered a potential removal rate. We have to understand that these floodplains are only active when inundation occurs. On average, at Shadow Ditch, the floodplains were inundated about 91 days out of the year during the 3-year study period. That means that on an annual basis, the two-stage ditch floodplains specifically remove about 940 kilograms of N per year and prevent them from moving downstream. To put this into context further of our annual watershed export, between 2017 to 2019, we estimate that about 46,000 to 55,000 kilograms of N left the watershed per year. And this is a lot of nitrogen. These were 3 of the biggest water years that we had at Shadow Ditch in the 12 years that we had been working there. And based on these estimates and what you saw on the previous slide, the two-stage ditch floodplains remove 2 to 3% of annual export. But we have some important factors that must be considered when assessing this relative percent removal. The first factor to consider is the floodplain bench width. When they constructed the upper reaches of the two-stage at Shadow Ditch, they decreased the bench width, which reduced the potential for removal by cutting it in half. Additionally, the flooding threshold is important to consider, making sure the benches are built low enough so that flooding frequency is high to promote nitrogen removal on the floodplains. And then finally, interannual variation in flow, which in turn drives annual variation in export, is also important to consider in determining the effectiveness of the two-stage ditch floodplains. Our results suggest that the two-stage ditch is an effective ecological engineering tool to reduce downstream nitrogen loss. But this watershed nitrate load reduction is dependent on both construction details, including bench height and floodplain area, as well as environmental contexts like interannual variation in flow, flooding frequency, and inundation thresholds. We also must consider whether lags in nitrogen removal post-construction could impact export during critical periods when implementing restoration. And because of this, we suggest that construction should occur during periods of low export like summer base flow to minimize impacts of construction. Finally, the floodplains should also be constructed to maximize their inundation frequency and floodplain surface area to enhance their functioning. All in all, our results suggest the two-stage ditch is an effective infrastructure enhancement in agricultural ditches that results in the improvement of ecosystem function, not only in reducing nitrate export, but also helping to increase sedimentation and improve stability of agricultural ditches, which can in turn mitigate risks to downstream ecosystems. With that, if you have any questions, I'd love if you contact me at my email up here on the screen, and keep your eye out at Ecological Engineering for our publication coming out soon. Thanks.